Hello, my name is Ed Chapman, and this videocast is going to cover the first part of a tour of the cell, a basic introduction to cell biology. It all started with microscopes about 1650, about halfway through the 17th century, when two scientists figured out how to take some lenses and magnify things in a way that made it clear enough and bright enough that we could see individual cells. So up until about 1650, the best you had was the unaided eye. Today we have both light microscopes and electron microscopes. And electron microscopes have incredible resolving power. They're able to produce clear, sharp images at very high magnifications. But the light microscope is pretty much what you need to study individual cells. Now, if you want to study the parts of cells, you need an electron microscope. There are two types of electron microscopes used at the college and university level. The transmission electron microscope produces slides that are very highly magnified cross sections of things placed on a microscope slide. And it works pretty much like a light microscope, except the magnification power is much higher, up to 250,000 times magnification. As you can see in this picture here, we have a Golgi apparatus magnified many times. Scanning electron microscopes, on the other hand, produce three-dimensional images by using an electron beam to scan the exterior of a three-dimensional mounted specimen. And this is how we get sharp pictures of insect parts and these bacteria. Both of these electron microscopes are used to study cells. Cell biologists also study cells by taking them apart. There are two processes involved here. The first one is chopping the cell up or homogenizing it into something called you could call it a cell smoothie in a way. And then the second part is separating the components by spinning or using centrifugal force to force very heavy parts to the bottom of a test tube and the lighter parts to the top of the test tube, and everything else between sorted by its individual density. It's a lot like what happens in a washing machine when the wash cycle's finished and the whole drum spins to force the water out of the clothing. Except in this case, everything stays in the test tube, and the heavier parts end up in a pellet, and the lighter parts end up in something called the supernatant, as you can see in this diagram. The most basic difference in cell biology is the difference between prokaryote cells and eukaryote cells. These are two very different types of cell architectures. Excuse me. Prokaryotes are the very small, simple, one-compartment cells that you guys know as bacteria. Eukaryotes, on the other hand, are larger, much more complex. They're made up of many different compartments. And that type of architecture, the eukaryote plan, is used to build plant, animal, and fungi cells. So, why are cells so small? Why don't we have cells the size of water balloons floating around in the ocean, for example? And the answer to that is just a law of physics. As you increase the size of something, two things are getting larger. Its surface area and its volume. But surface area increases by the square, whereas volume increases by the cube. And if you think back to geometry about how you calculate area versus how you calculate volume, you'll see this. So what this means is as a cell grows larger, the amount of surface area per unit volume, or the ratio between surface area and volume, decreases. So that means that if a cell's doing most of its metabolism using its membranes, as it gets larger, it has to do more and more metabolism in a more and more restricted area. So the net result of this is single large cells cannot be very metabolically active. So in order to be metabolically active, they have to increase their surface area. And they do this by dividing and becoming smaller. If you look at these frog cells, frog eggs start out as fertilized zygotes growing inside of a jelly-like coating. And if you look in the top picture here, you can see that dark, sort of planet-looking fertilized egg cell there floating in a bunch of yolk surrounded by a jelly matrix. And as that little tadpole starts to develop, the volume of that cell gets divided up between increasingly smaller and smaller cells until finally they're microscopic, and you can start seeing tissues and organs, like in the lower picture where you can see the eyes, the brain, which is like the lightest part, and even some blood vessels. So life on Earth, multicellular life on Earth, solves this problem of surface area to volume by becoming multicellular and making lots and lots of little cells all stuck together with lots of surface area. Compartmentalization is the theme in eukaryote cell biology. If you look at this diagram here of an animal cell, you can see lots and lots of compartments, lots of rooms, lots of places where things can happen. So eukaryotes have evolved compartments to specialize functions in certain areas. For example, respiration is specialized in mitochondria, and manufacturing for a cell is specialized in something called the Golgi apparatus in the ER, which we'll talk about a little bit later. But because each activity is limited to one place, it can happen much more quickly and use much less energy. Specialization is an energy-saving evolutionary pathway. The rest of the slideshow, we're going to talk about the cell parts that you should know and what their function is. So we're going to focus on structure and function, the connection between these two ideas. That's going to be the theme of this chapter. Let's start with probably the most familiar part of any cell, going all the way back to probably elementary school, is the nucleus. Everybody's heard that the nucleus is the, quote, brain, unquote, of the cell. That's kind of a simplistic way of looking at this. We're going to have to be a little bit more technical here. The nuclei of eukaryote cells are the place where the cell is storing its chromosomes. So you can think of chromosomes as being the genetic library or the genome of a cell. And in addition to being the genetic library, the nucleus is also where ribosomes are manufactured. And we'll talk more about ribosomes in a few minutes. The genetic library, which is made of mostly DNA, is referred to as chromatin when it's not dividing. And when a cell starts to go into cell division, the chromatin changes its form. It becomes visible chromosomes, which can be counted under a microscope. But until that happens, we just refer to this material as chromatin. And the ribosome factory area, so to speak, is called the nucleus or the nucleolus, and it's a darker area in a stained nucleus or nucleus slide. Ribosomes, as I said, are the ribosome factories for cells, and of course ribosomes are important in protein synthesis. Another name for ribosomes is rRNA, little rRNA, which means ribosomal RNA. And it's the place in the nucleolus where DNA is being transcribed into material that's going to become ribosomes. It's not going to become messenger RNA or tRNA. And the job of ribosomal RNA is to interact with transfer RNA and messenger RNA, which you might remember from biology. All three types of RNA combine to work together to build polypeptides, which are proteins when they get folded up properly. So let's look at a picture of this. If you look at this diagram and we label the parts, the messenger RNA is this strand here, which actually comes into the ribosome from this side, travels through the groove, and comes out this side. And as it travels through the groove, the information, which are codons, 3-unit letters of nucleotides here, are translated into amino acids. And there's a tRNA player in this activity also that's not shown in this picture. So ribosomal RNA, messenger RNA, and finally tRNA work together to produce this strand of amino acids, which is going to become a protein. All right, that's the end of part 1. We will pick up with part 2 in the next video cast. Hope you enjoyed this, and thank you.